after the spinach pigments are isolated and analyzed. The components of the pigments can be separated and isolated using column chromatography. In this experiment, the carotenes and chlorophylls are separated and isolated from the rest of the pigments. Similar to the TLC technique, the components of the spinach pigments are separated in a column of adsorbent material, such as silica gel, due to the difference in the polarity. The stationary phase is the adsorbent material such as silica gel packed in a column, typically a glass column. The mobile phase is the eluent to move the sample through the column. Usually, multiple solvents or mixtures are used. In a silica gel column, a higher polarity compound will bind stronger to the adsorbent, compared to the lower polarity compound. Therefore, a higher polarity compound will move down the column slower, when it is eluted by a less polar solvent, compared to the lower polarity compound. Here is an illustration of separation in column chromatography. The red compound is moving down the column faster, compared to the green and blue compounds. The red compound is the least polar, while the blue compound is the most polar. To prepare the column, a glass chromatography column is packed with cotton, sand, the silica gel slurry, and then another layer of sand. 3 solvents are used: hexanes, 95-5 mixture hexanes acetone, and a 50-50 mixture hexanes acetone. The pigments and eluents are added to the column slowly using a pipette. In the experiment, we will see the first yellow band is separated from the rest of the pigments, when the pigments are eluted using a lower polarity solvent. The carotenes are separated and isolated from the other pigments. To separate the chlorophylls, a slightly more polar solvent is used, 50-50 mixture of hexanes and acetone. After the carotenes and chlorophylls are separated and isolated from the spinach pigments, another TLC analysis can be done to evaluate if the separation of these two pigments from the rest of the pigments are successful.